Good afternoon, everyone. My name is Dr. Angie Ambers, and I'm the Assistant Director of the Henry C. Lee Institute of Forensic Science in West Haven, Connecticut. And I'm going to be talking to you today about a post-conviction case in Texas, a wrongful conviction for murder in Texas, and how we used probabilistic genotyping to analyze the DNA evidence and help exonerate this individual. So probabilistic genotyping is a kind of a new tool in our tool set for forensic DNA analysts, and it really helped reduce the subjectivity in the way that we interpret DNA mixtures in criminal cases. So this case pertains to the conviction of a man named Lydell Grant for murder in Texas, and this is Lydell when he was arrested for murder in Houston, Texas, and primarily the evidence that was used against him during his trial, and I'm going to go through a background of the case, was eyewitness identification and flawed DNA mixture interpretation. And I say faulty eyewitness identification because as anybody that studied criminal justice and eyewitness identification, we know that it's notoriously unreliable, especially when the eyewitness, the race or ethnicity of the eyewitness and the race or ethnicity of the perpetrator are different. And in this case, the perpetrator, Lydell, is obviously African American and the eyewitnesses were Caucasian. And then flawed DNA mixture interpretation. This is not the actual DNA mixture that was used in this case, but for those of you that are not familiar with what a DNA mixture looks like on an electropharogram, so this, if you look on the far left-hand side of the screen under Mr. Grant's photo, you see the DNA locus or DNA marker name D3S1358. That's one of the DNA markers that we analyze, and across the top of this, across the line where D3S1358 is, you see VWA, D16, CSF, etc. Those are the names of the DNA markers tested. And then if you look under each DNA marker name, you see several peaks with a number underneath each peak. And in a single source sample, a DNA sample from one individual or from one person, you would only see two peaks or one peak under each DNA marker. Because again, humans are diploid organisms, so we have two alleles per locus or per marker that we test. If you have two different alleles at a particular DNA marker, you would see two peaks on the electropharogram, and if you have two of the same alleles, if you're homozygous for that marker, you would see a single allele. So obviously in this image, I'm showing you an example of a DNA mixture. There's far more than two allele peaks underneath each DNA marker that's tested, and that's because there's more than one individual DNA present in that mixture. And this type of scenario of a DNA mixture is what was part of some of the key evidence used to convict Mr. Grant. So just a little bit of background on the case. This is Aaron Scheerhoorn. He's the stabbing victim in this case. And the murder, again, happened in Houston, Texas, in an area of Houston called the Montrose district. And it basically is like a nightclub, bar, restaurant district in Houston, and it's particularly well known for being a club and bar district for the homosexual community in Houston. So when Mr. Scheerhorn was murdered and stabbed to death in the parking lot outside of this bar, a lot of, the media got a hold of it, and of course they were calling it a hate crime because of his sexuality and the area of town where he was killed. So essentially, Mr. Scheerhorn was not actually inside the club. He had been out and about in the Montrose district that night in December 2010 And he had encountered an African-American male. They got into a fight. He had been stabbed multiple times at that point. He got away from the perpetrator and was running from him. And he actually ran up to the door of Club Blur, as you can see in this image. And there were two bouncers standing at the door, and he begged them. He lifted up his shirt to show the bouncers that he had been stabbed, and he was begging them to let him inside for sanctuary, to be protected because this perpetrator was still chasing him. The bouncers did not let him inside the club, and while they were arguing about letting him in, the perpetrator caught up with Mr. Scheerhorn and proceeded to stab him to death in the parking lot of the club. And there were several eyewitnesses that were standing around in the parking lot outside of Club Blur, and they witnessed the stabbing. And I will say that, one of the things that disturbed me about this case, other than that this man had clearly been stabbed in his abdominal area and they wouldn't let him inside the club for safety, is that there were a half dozen or so eyewitnesses that watched him get stabbed to death essentially in the parking lot, and no one tried to intervene and help him, nor did they try to detain the attacker. So the attacker ran off and was never caught. They did call 911, and of course Mr. Scheerhorn died shortly thereafter. So then how does Lydell Grant come into play here? Because I told you that the witnesses did not try to detain the perpetrator, nor did they try to help Mr. Scheerhorn. So about a week later after the murder occurred, one of those eyewitnesses that watched the stabbing was at a restaurant, a cafe in the Montrose district in Houston, and saw our client, Lydell Grant, and thought, that looks like the man that I watched stab Aaron Scheerhoorn in the parking lot of Club Blur last weekend. So this individual followed Lydell Grant out into the parking lot, snapped a photo of his license plate number, and then reported that to the Crime Stoppers Anonymous tip line. They basically, this individual basically called Crime Stoppers and said one of the eyewitnesses to that murder that happened in the Montrose district, and I just saw the perpetrator, and here's his license plate number. And so long story short, that's how Lydell kind of got intertwined or even on the radar of the police for this case. So after they put out a warrant for Lydell, arrested him, and charged him with the crime, or were in the process of charging him for the crime, they did a police lineup, and the eyewitnesses that watched the murder occur in the parking lot outside the club, all 6 eyewitnesses identified Lydell as the perpetrator. So in addition to the eyewitness identification, during the victim's autopsy, they collected the fingernails of the murder victim because the eyewitnesses had said that he struggled, he fought for his life. He was stabbed multiple times during this altercation. And ultimately, as you can see here, this is a screenshot of the DNA mixture that was recovered from the fingernails of the murder victim during autopsy. And so part of the case was, okay, we've got the 6 eyewitnesses that have identified Lydell Grant as the perpetrator, and then the prosecutor wanted to know, is the DNA mixture that was recovered from underneath the fingernails of the murder victim, is that a mixture of the victim and Lydell Grant? Is Lydell Grant present in that DNA mixture? And this is actually a screenshot of the final case file report, the DNA summary report that was used during Mr. Grant's trial in 2011. And item 12.2.1.1 was a swabbing of the murder victim's fingernails, and as you can see, they're reporting that it was a DNA mixture, that Aaron Scheerhoorn cannot be excluded as a contributor to the mixture, which, you would expect, because it was his fingernails, you would expect his DNA to be present. And then they've applied a statistic to that inclusion. And then, of course, highlighted in yellow at the bottom, again, this is the final report that was used during Lydell's trial. It says, no conclusions will be made regarding Lydell Grant as a possible contributor to this DNA mixture. So the analyst, the DNA expert for the state in this case, basically said, this DNA mixture is too complex. I'm not going to attempt to draw any conclusions, so I'm going to call it inconclusive. We're not going to say Lydell Grant's DNA is present underneath the fingernails of the victim, but we're not going to say he's not present either. We're just going to say inconclusive. The reality is, this is essentially, I think, this is the conservative stance that should have been taken. It should have been an inconclusive result. However, the way that it was presented, the actual testimony of the expert during the trial was a little questionable. So when the prosecutor was asking the DNA expert, were you, in front of the jury, were you able to identify Lydell Grant's DNA underneath the fingernails of the murder victim, she said, I wasn't able to do that. I called it inconclusive. The DNA mixture was too complex, which was fine. But then the prosecutor went a step further, and I'm paraphrasing some here, but it was something to the effect of, oh, well, you can't say he's not in there then, correct? And she basically agreed with that. And so it kind of left the door open in the minds of the jury that, oh, well, she's not saying Lydell Grant's DNA is not there. It could be there. Could very well still be there. And then you add that on top of the fact that there were 6 eyewitnesses that testified and visually identified Mr. Grant, and as you can imagine, that was enough to seal his conviction. So again, you've got 6 eyewitnesses that are saying Lydell Grant was the man that stabbed Aaron Scheerhoorn to death. Now, at this point, when I'm reviewing the case logs, I was the DNA consultant on this case, part of me also was wondering, other than those of us that work in the criminal justice field know that eyewitness identification is flawed, it's not very reliable. The other thing that popped into my head here was, this was a murder that occurred in a parking lot of a bar on a Saturday night. And so these eyewitnesses very likely probably were under the influence of alcohol. So if you tack on the intoxication, the alcohol intoxication on top of just the general unreliability of eyewitness identification to begin with, that's one of the things that makes this even more troubling, that Mr. Grant was convicted solely on eyewitness identification by eyewitnesses that likely were intoxicated at the time they were witnessing the crime. So I got involved in this case as a consultant in early 2019. So Mr. Grant was convicted again, I'll remind you, in 2011. So he had already been in prison for several years. And during the time that Mr. Grant was imprisoned, there was quite a bit of study being done at the federal government level and by independent groups of experts, where we were looking into all the different disciplines in forensic science and assessing the reliability and the validity of the methods that we were using in criminal trials. And in 2009, actually, this was obviously a little bit before Mr. Grant's conviction, there was a report called Strengthening Forensic Science in the United States at Path Forward that was published by the National Research Council and the National Academy of Sciences. And it basically kind of was an overview of the different disciplines and the current state of the different forensic science disciplines and how we could improve upon our current methods. And then in 2016, September 2016, there was a pretty influential publication that came out. It's called the PCAST report, and PCAST stands for the President's Council of Advisor on Science and Technology. And this was a council of experts that were put together, scientific experts that were put together under President Obama's administration. Again, building upon what the NRC and National Academy of Sciences report said. It basically said, you know, we've got room for improvement in our forensic science methodology. And this PCAST report, you know, it's a pretty long report, but as far as how it relates to this current case, one of the areas of weakness that they identified as far as DNA, forensic DNA analysis, is in how we interpret forensic DNA mixtures. So the report was basically saying that our forensic science, our forensic DNA technology, the instrumentation we're using, the laboratory methods that we're using to generate the results are valid and reliable, but where we could stand for some improvement is in how we interpret the data that we've generated in the laboratory. And so, you know, I've shown you a screenshot here. One of the statements from this report was complex DNA mixture analysis needs additional support to be deemed scientifically valid and reliable. And, you know, this was late 2016. Again, Mr. Grant's already in prison for murder. And they just keep, this PCAST report keeps getting a lot of press. And so I've highlighted down at the bottom of the screen here, you know, this study aimed to help close the gaps for a number of what are called forensic feature comparison methods. So again, this report was not just an overview of the current state of forensic DNA analysis, but a lot of other forensic science methods, like how we compare DNA samples, bite marks, latent fingerprints, firearms and ballistics, footwear patterns, hair analysis, etc. And then right after this PCAST report came out, Forensic Magazine, which is geared towards a lot of people that maybe aren't working within the forensic science field, but that want to keep abreast of all the current and latest developments. You know, Forensic Magazine published a huge article that says subjective DNA mixture analysis, which has been used in thousands of cases across the U.S., has been blasted by a White House panel. And again, that White House panel is that President's Council of Advisors on Science and Technology, the PCAST Council. And basically, both the PCAST report and this particular article, what they said was, again, that when we as individual trained forensic DNA analysts try to interpret a DNA mixture that's recovered from a victim or a crime scene, there's some level of subjectivity that goes into that analysis and that interpretation, and that we need to find a better way, a more objective way to analyze these DNA mixtures. And one of the suggestions was that we move more in the direction of artificial intelligence, and we allow computers, specifically computer software programs, to analyze these DNA mixtures rather than us individually as people trying to analyze these mixtures to reduce, again, that subjectivity. So in the process of that PCAST report being developed, remember that report was published in late 2016. There were some other things that were going on as a result of that 2009 NRC report. And one of the things that we were doing was auditing forensic science laboratories, criminal crime labs across the U.S., and kind of assessing how things were being done in these laboratories and is it scientifically sound, reliable, valid, et cetera. And one of the things that made national headlines in April 2015, so about a year before the PCAST report came out, was that there was a big lab in Washington, D.C., a cream of the crop type lab, a high-end, high-level lab that a national accreditation board found flaws in the way that they were interpreting DNA mixtures. And essentially in the bottom right hand of the screen, if you read this, all of the casework testing that was being done by that lab. Once this audit was done, the lab was basically shut down and all the casework was suspended, because what they found was that the problems with that lab had centered on the analysis of evidence that, again, includes DNA that's from more than one person, and the conclusions regarding the likelihood that a certain person's DNA is included in that DNA mixture or not. So again, we're further illuminating as this process is moving along that there are some issues with the way we were interpreting DNA mixtures. After this Washington, D.C. lab was shut down and casework operations were suspended, the state of Texas, which is where I was working during this time period, then said, whoa, basically if they're doing this incorrectly, if there are weaknesses in the way DNA mixture interpretation is being done in Washington, D.C., I wonder if we have issues in the state of Texas as well. So there was this statewide audit that was done of all the DNA labs in Texas, and they wanted to look at, how many cases have already been adjudicated and how many current cases do we have where a DNA mixture is a key part of the evidence that either was used in the original conviction or acquittal or is currently being used for the current probative cases. And what they found was that since 1999, and again, this was 2015, there were over 25,000 cases that were tried in Texas courts that involved DNA mixtures. And so they actually sent a letter to all inmates, or maybe they weren't inmates anymore and they had been released, but anyone that had been convicted of a crime in Texas where a DNA mixture was involved in their conviction. And they sent them letters and basically said this, we've since identified that there are issues with the way, subjectivity issues with the way DNA mixtures are interpreted, and this hence may be grounds for an appeal for you or a new trial. And so this was huge in the state of Texas. Again, here's another article that was published in Texas reviewing thousands of DNA cases that used outdated methods for calculating odds. And again, this was August 2016, about a month before the PCAST report came out. But they were building on the Washington, D.C. audit and the 2009 NRC report. And, we just realized with interpretation comes subjectivity, and anytime there's subjectivity involved in interpreting a DNA mixture, we can make errors. We can potentially, prosecute the wrong person, convict the wrong person, or even possibly the flip side of that is acquitting the wrong person. So as far as subjectivity, with this big statewide audit in Texas, what we found was that different DNA labs across the state of Texas were using different methods or different approaches to interpret DNA mixtures recovered from crime scenes. There was a lack of standardization as far as how to go about analyzing and interpreting a DNA mixture. The obvious, like in murder cases such as this case and maybe rape cases, you would expect the victim's DNA to be present in the mixture, and logic would tell you can subtract the known DNA profile of the victim out of the DNA mixture and therefore help deduce the profile of the perpetrator. That was not necessarily being done across the board throughout labs in Texas. And then part of this statewide audit was to take complex DNA mixture cases from various labs across the state of Texas and make copies of the DNA mixtures that were used in those trials, disseminate those out to several different highly qualified DNA analysts and ask them to independently review and interpret the mixtures. And what we found was when you give a copy of the same DNA mixture to several different highly competent DNA analysts, many times the different analysts independently would come to different conclusions about whether someone was included or excluded from that mixture. So obviously, just as an example here, I'll show you one of the things that we were doing before probabilistic genotyping is we were doing interpretation of DNA mixtures based on what we call the major contributor, minor contributor scenario, where the height of the DNA peak in an electropherogram is correlated to the amount of DNA present. So in a lot of these cases where you would expect the victim's DNA to be present under their own fingernails, or if it's a rape case where it's a vaginal swab, you would expect a lot of the victim's DNA to be present, and therefore the peaks would be higher, and maybe less DNA from the perpetrator would be present, and that would be your minor contributor. And so if we were looking at a DNA mixture and we thought visually this is a clear major minor contributor scenario, we were again trying to manually interpret that mixture. And in this case, if you look at this slide, I've got marker 1. So this is showing you an example of an electropherogram for 4 different DNA markers tested. If you look at the 2 peaks under marker number 1, many analysts would interpret this as being one person in this DNA mixture has 2 copies of allele A. And under marker 3, because of the height of the peak, this person has 2 copies of allele B. Because recall from earlier in this presentation that when you have 2 of the same alleles, that peak sits on top of each other and it increases the height of the peak because there's more copies of that allele present. So many analysts would, interpret that part of the mixture. Under marker number 4, many analysts would say, well, the height of the peaks for allele A and allele D are higher, so that's clearly the major contributor. And the peaks for the allele B and C under marker 4 are the minor contributors. So that's how they would pair the alleles together and separate the mixture into DNA profiles for 2 individuals. But, this can get, again, when you have multiple different people looking at the same DNA mixture, you can arrive at different conclusions. There's some subjectivity. So for the second marker, many analysts would say, okay, one individual in this mixture for this marker has an allele A and an allele C. For marker number 3, same thing. Some analysts may say the major contributor is BB and the minor contributor is AC. For marker 4, again, a lot of analysts would say there's a clear major minor contributor there. But then you go back over to the first marker on the far left-hand side of the screen, and even though most trained DNA analysts would have agreed that the major contributor in this scenario has 2 copies of allele A, when they were deducing the allele pairings for the minor contributors, some analysts may say they have 2 copies of the B allele, and other analysts may say, well, I think they have 1 copy of the A allele and 1 B. And then if you look underneath the second marker, there's several different scenarios there that different analysts could arrive at as far as what the allele pairings are for the minor contributor. So, and of course, keep in mind that in standard casework, we're analyzing now 20 different DNA markers, and you're going to go through this process for each marker in the DNA profile. And so it's a lot more than just the 4 markers that I've shown you on the screen. In other scenarios, so on the previous slide, I was explaining to you kind of what we considered in the field a clear major contributor, clear minor contributor scenario, or relatively straightforward. But the reality is that's not what a lot of mixtures were looking like in criminal cases. You might, in other words, see something like this where the peak heights are pretty similar between the peaks for each marker tested. And so there's no discernible major minor contributor. So at that point, and I'll go back to that slide, it gets even a little more complex in how you're subjectively determining which 2 alleles pair with which person for each marker tested. So what the NRC report and the PCAST report basically said is that we need a better way to do this. We need a better way to interpret complex DNA mixtures. and there's too much subjectivity involved with people, no matter how well trained you are, how intelligent you are. So let's let computers do it for us. And so there were software programs developed to help interpret complex DNA mixtures, and these software programs do what's called probabilistic genotyping, wherein, and I'll go back to this previous slide, the software programs are looking at peak heights and various key components of the electropherogram and calculating the probability of each allele pairing up with another allele to deduce the proper, what the highest probability is that of which allele should be paired together in this mixture, so that you can deconvolute the mixture of DNA profiles into separate individual profiles. And two of the big probabilistic genotyping software programs that are currently being used in casework labs, one is called StarMix, and the other one is called TrueAllele. And they basically both do the same thing. They both are interpreting DNA mixtures based on probabilistic genotyping. And again, it's the less subjective. We can never completely reduce the subjectivity in DNA mixture interpretation because, again, it's a blind process. You know, we often say that forensic science is a historical science because we are, as analysts, we're actually analyzing the remnants of a crime. We're not actually present when the crime's being committed, when that DNA mixture is being formed. So we have to analyze the data after the fact, and that's what these software programs do. And again, the less subjective approach is what we were shooting for because we're also dealing with some unconscious bias sometimes with human analysts. But the previous way we were doing it, again, involved calculation of what's called a CPI or CPE, combined probability of inclusion or combined probability of exclusion. We have abandoned that approach now, again, and we're using these computer software programs. So we still have the human DNA analyst that is extracting the DNA from the evidence, doing all of the laboratory work to generate the DNA profiles, but once the DNA mixture profile is developed and produced, then we're letting a computer software program analyze the data instead of us doing it as humans. So I'll take you back to this previous slide in the presentation where recall that at the time of Mr. Grant's original trial in 2011, they had a DNA mixture that was recovered from the fingernails of the murder victim, and they were not able to draw any conclusions about whether Mr. Grant was included or excluded from that mixture. So they just called it inconclusive. And so in February 2019 is when I started consulting on this case, and of course I'm familiar with the NRC report, the PCAST report, et cetera. So I thought, okay, well maybe, you know, this technology was not available to interpret DNA mixtures at the time of the trial, but let's see what these probabilistic genotyping software programs will say now about whether Mr. Grant is included or excluded from that mixture. So before I sent the raw data that was originally generated back in 2011 for probabilistic genotyping analysis, I thought, let me just do kind of a quick screen. Let me take that DNA mixture that was produced from the evidence in 2011 and let me just list the alleles in a basic Excel spreadsheet. And this is what I did. So just kind of at first glance, what does it look like? Could he be possibly in this mixture? Could he not? And you'll see here, so on the far left hand of the screen, you'll see a column called locus. That's the name of the DNA marker that was tested. You've got Lydell Grant's individual DNA profile that we obtained from his reference sample. We've got in the third column, the victim's DNA profile from their reference sample. And then the subsequent 6 columns are the alleles that were present in that DNA mixture. And what I wanted to see was, obviously, we would expect the victim's DNA to be present in there, but is are there some common alleles with Lydell Grant? And what I started seeing was just on this initial screening, of course, this is not the way we analyze DNA mixtures, but this is kind of a first glance approach. It was pretty obvious to me that Mr. Grant, his DNA is not present in this mixture. So everything that's highlighted in yellow here for you and in red color are alleles that were present in the mixture, the DNA mixture that was recovered from the victim's fingernails, that do not correspond to alleles in the victim's DNA profile or Mr. Grant's DNA profile. And there were 26 what we call foreign alleles that were present underneath the murder victim's fingernails that don't match the murder victim or Mr. Grant or the suspect, the guy that was convicted of this crime. So I should point out here that this is something that took me about 30 minutes to do that could have very easily have been put together and shown during Lydell Grant's original trial in 2011. But I'll point out that Lydell Grant was an indigent client, an indigent defendant. He had a public defender defending him, and that public defender did not hire a forensic DNA expert on Mr. Grant's behalf. So all the jury heard was what's on this previous slide, that there was a DNA mixture recovered from the murder victim's fingernails, and the DNA mixture is too complex, we can't interpret it, so we're going to call it inconclusive. But then, of course, they heard that there were 6 eyewitnesses that identified Mr. Grant. Now, with that being said, back to this slide, again, this took me 30 minutes to put together. I think even in 2011, before we were using these computer software programs to analyze DNA mixtures, had Mr. Grant's attorney hired a DNA expert, this might have, and they had posted this up on the screen so the jury could see it, this might have planted that reasonable doubt in the minds of the jury that they had whether or not they had the correct person. This might have been enough to plant that reasonable doubt. And remember, the burden of proof in a criminal trial is you have to think that someone is guilty beyond a reasonable doubt. There's clearly someone else present underneath the fingernails of the murder victim just by looking at this basic screening tool. So I was working with Mike Ware, who's the director of the Innocence Project of Texas. He's a defense attorney. So I called him. He's in Texas. I'm in Connecticut. And I send him a screenshot of this. And I'm like, well, you're right. I don't think Lydell is in the mixture. But what we need to do, based on the current recommendations of the PCAST report and all of the national audits that are happening, is we can't just go with this. We need to now send this raw data, this DNA mixture data, and have it analyzed with one of these probabilistic genotyping software programs. So we sent the raw data to a company called Cybergenetics in Pennsylvania, and this company is the developer, the maker of True Allele DNA interpretation software. And what you can do, so this was March of 2019, you can submit the DNA data that contains the data for the DNA mixture as well as the DNA profiles of the victim and the suspect, and they will run it through their probabilistic genotyping software, and they will send you back a report and tell you, based on their software analysis, who's included in the mixture and who's excluded. And you get it in an email response. And basically, you can see here, this is actually a screenshot of the email that I got back in March 2019 saying, if you look at the highlighted region, victim Aaron Scheerhoorn's DNA was found on his fingernails. Well, we knew that because they are Aaron Scheerhoorn's fingernails. But the very next bullet point there, you'll notice, is defendant Lydell Grant's DNA was not found on the victim's fingernails. So he's excluded as a contributor to that DNA mixture. And then they go on to state DNA from an unknown person was found on the victim's fingernails, and they, again, subtract the victim's DNA profile out of the mixture because you know that person's DNA is in the mixture. And they essentially, the software program essentially deduced the alleles that are in the DNA profile of the foreign contributor. And so we got excited at that point. We thought we knew we were moving in the right direction and that they had indeed convicted the wrong person. But on top of the email that you saw in the previous slide that states who's included and who's excluded from a DNA mixture, one of the other things that cybergenetics will do is they apply a statistical weight to that inclusion or exclusion. And you can see item 12.2.1.1 is the victim's fingernail. We also had a DNA profile from the victim, a reference sample, a DNA profile from Lydell Grant, the accused, and they apply a statistical probability. So you can see this is the exponent, that 19 underneath victim. That's the exponent for the statistical value. So you can see this is somewhere between, obviously the victim's DNA is included somewhere up in the quintillion, septillion numbers. He's obviously included. We expected that. But look at Lydell Grant's reference DNA profile. He is excluded between the billions and trillions. There's 8.5 billion individuals on planet Earth, so this exceeds the human population of Earth. And then the unknown male contributor who's present in that DNA mixture, both that unknown male contributor and the victim are determined to be the two individuals in that mixture. They're included in the mixture. Lydell Grant is excluded. So we thought at that point that, that was our case, that was our grounds for appeal. We've proven that Lydell Grant's DNA is not underneath the fingernails of the murder victim, and we contacted the Conviction Integrity Unit at the Houston DA's office. We wanted to move forward with the appeal, and then the thought process became, well, so what? Aaron Scheerhoorn was out in the bar district on a Saturday night in Houston. He's a gay man. He might have had a sexual encounter earlier that evening, and that foreign contributor, that unknown DNA profile underneath his fingernails, might not be related to the murder at all. It might be related to a sexual encounter that he had earlier that evening. So we knew we had to find out who this DNA profile belonged to. So in April of 2019, we worked with John Donahue, the Beaufort County Sheriff's Office in South Carolina, to have this unknown DNA profile that was found in the mixture searched against the FBI's CODIS DNA database. And we got results back a couple of months later in June 2019 that we had a hit in the CODIS database. And let me remind you that the CODIS, the national DNA database maintained by the FBI, is vastly incomplete because it only contains the DNA profiles of convicted offenders, so people that have been caught, and it contains, for some states, it allows us to profile the DNA of all arrestees, so people that have been arrested but not yet convicted. So anybody that hasn't been arrested and hasn't been caught and convicted will not be in this database. So there's a chance that we could have not had a hit at all or a match in the database at all. We did get a hit to an individual named Jermerico Carter, and not only had he been in and out of the prison system throughout his life, he's a habitual criminal, but we did some background investigation on him. He was known to not be incarcerated at the time Mr. Scheerhorn was murdered. He lived in the Houston area where Mr. Scheerhorn was murdered during that time period, and he moved away from Houston a couple of months after the crime occurred. He was actually tracked down in December in Atlanta, Georgia, and arrested for the murder of Mr. Scheerhorn, and he has actually confessed to the crime. So June 2019 is when we identified Jermerico Carter as the perpetrator, and it took us several more months after that to get Lydell Grant out of prison. So this is an image. In the top image, on the far left, that's Mike Ware, the director of the Innocence Project of Texas. And you can tell Lydell is happy in this photo. This was right when he found out he was going to get released from prison. This was the day before Thanksgiving, so late November 2019. And then the bottom photo is him walking out of the courthouse after he's been released from prison. And that's his mother and his brother that he is with. And then, so that, again, he was released from prison the day before Thanksgiving. And then December is when they finally tracked down. At this point, we were going to be preparing for a new trial for Lydell, so we could present all the new DNA evidence, probabilistic genotyping analysis, and all the CODIS hit and everything. And in the meantime, the police were searching for Jermerico Carter that we identified in the FBI's database. And these are some screenshots of some articles that came out. He was arrested, new suspect arrested, charged in 2010 Montrose stabbing, wanted murder, hid in Atlanta for years while an innocent man sat behind bars. And then I just wanted to show you an image here. Again, on the left, this is Lydell Grant's mugshot when he was arrested in 2010. And on the far right is Jermerico Carter when he was arrested in December 2019. And, you can study, you can go back. There are multiple instances throughout history of where DNA has been used to exonerate someone that was originally convicted based on eyewitness identification. And if you end up looking at an image of the convicted person and the true perpetrator that was later convicted, there do tend to be some similarities in the way they look. As far as this happening with wrongful convictions, you can look at the National Registry of Exoneration. Since 1989, there have been 2,611 exonerations, and the combined imprisonment time, more than 23,000 years lost, the people that were wrongfully convicted. And I'm not going to spend a lot of time on all of these stats, but this graph here talks about the various contributing factors to wrongful convictions. And I've particularly circled mistaken witness ID, which was a factor in Lydell's wrongful conviction, and false or misleading forensic evidence. Again, there wasn't false evidence in this. It just was that there was a DNA mixture recovered, and it was called inconclusive, and the way it was presented in court was misleading and kind of left the door open for the jury to interpret that he possibly could be in the mixture. And then again, compounded with the 6 eyewitnesses that identified him, that kind of sealed his fate with that conviction. And so I just want to point out here lastly that, the NRC, the 2009 NRC report and the 2016 PCAST report were geared towards, they're not pro-defense reports. They are definitely pro-science, pro-justice reports. And our goal in the justice system, whether we are attorneys or forensic scientists, should be to find the guilty and protect the innocent. We don't want cases like this to happen where someone spends 10 years in prison for a crime they didn't commit. So, we've had this mindset. We need to go back to this. There's a 19th century medical legalist that said, if the law has made you a witness, you must remain a man of science. You can't go into it thinking you have a victim to avenge or a guilty or innocent person to convict or save. You have to bear testimony within the limits of science. I want to dedicate this to a lot of people. You know, they hear about these cases and they think, oh, you're just working for the defense. You were working for Lydell Grant, the convicted, the accused. But ultimately, you know, this is a pro-justice stance that we took on this case. So I'd like to dedicate not just the work that we did to this case, but this presentation not only to Lydell Grant, who was wrongfully convicted, but also to Mr. Scheerhorn, the victim, because we've finally been able to get justice for him and his family and identify the person that really committed this crime. And this was a joint effort. You know, I'm presenting this as a DNA consultant on this case, but I by far did not work by myself on this. This was a collaborative effort between myself, and again, I work for the Henry Lee Institute of Forensic Science in Connecticut, the Innocence Project of Texas, and then the Justice Through Science symbol that you see here on the screen is one of the logos for Cybergenetics, the company in Pennsylvania that developed TrueAllele software. And again, this is Mike Ware. This was the team that worked on Lydell and Mr. Scheerhorn's behalf to make sure that justice was done in this case. Mike Ware, again, is the director of the Innocence Project of Texas, and he also runs the Innocence Clinic at the Texas A&M School of Law. And on the far right of your screen is Dr. Mark Perlin, who's the Chief Scientific Officer and the Chief Executive Officer of Cybergenetics in Pennsylvania, and he's one of the developers of TrueAllele technology, which again is the computer software one of the computer software programs that we now use to analyze complex DNA mixtures, and it has greatly reduced the subjectivity in interpretation. So with that being said, thank you very much for listening and for attending, and have a good rest of your day. Thank you. If you have any questions, feel free to reach out.